Introduce Dr. Lauren O'Connell, Assistant Professor of Biology at Stanford, as our genetics seminar speaker. Dr. O'Connell started at community college and then transferred to Cornell University, where she obtained her Bachelor of Science in Biology. Then she joined the Cell and Molecular Biology program at University of Texas at Austin as a graduate student. Following her initial work with Dr. David Cruz, Lauren worked with Dr. Hans Hofmann on the evolution of the social behavior network across vertebrate species. She uncovered circuitry in cichlid fish homologous to the mammalian social behavior network. In addition, she performed a comprehensive evolutionary reconstruction of the vertebrate social decision-making network, comparing neurochemical gene expression profiles across 12 brain regions in 88 species, revealing remarkable conservation. Following her PhD at UT Austin, Lauren received a Bauer Fellowship from Harvard University to start her own research group in the Center for Systems Biology, where she developed poison frogs as a model system to study genetics and environmental factors that contribute to biological diversity and adaptation. She has recently joined the Department of Biology at Stanford as an assistant professor. Dr. O'Connell's contribution to science is recognized by numerous awards, including the L'Oreal USA for Women in Science Fellowship, the Frank A. Beach Award in Behavioral Neuroendocrinology, the National Science Foundation Career Award, and the NIH Director's New Innovator Award. Additionally, Dr. O'Connell is a passionate and dedicated advocate for underrepresented minorities and women. She received the L'Oreal USA Changing the Face of STEM Mentorship Award, which is used to broaden participation in science through outreach. She developed the Frogger School program, a partnership with K-12 science teachers to bring engaging learning experiences to classroom. Her lab hosts interns through the Rising Interest in Science and Engineering Summer Internship Program for high school students, a Stanford initiative designed for low-income students and those who will be the first in their family to attend college. The education component of her NSF Career Award also includes creating a community college internship program. I went to Lauren's seminar at Illinois a few years ago. Her enthusiasm is infectious and inspiring. Her lab has made impressive progress over the past years. As an outstanding faculty, she will bring a wonderful seminar today. Thank you, and let's welcome Lauren.
Thank you, Lily. That was a very kind introduction. For those of you who don't know, Lily did an internship in my lab at Harvard like 4 years ago. So it's been a pleasure to watch her grow as a scientist. And so I want to thank the grad students for inviting me to give a talk. There's like no bigger honor than being invited by graduate students. So thank you. And it might be genetics talk, and I'm going to bounce around with ecology and evolution with some molecular stuff too. So I hope you at least enjoy the ride. Before I get started, I'm sitting in my apartment near Stanford, and I just would like to acknowledge that Stanford resides on the unceded land of the Muwekma Ohlone people. And my group does a lot of international field work, and this expands the communities to which we are accountable. And so our fieldwork is conducted in collaboration and on the unceded and ancestral lands of the Owa, Chachis, Saichila, Epera, and Yumbos peoples of Ecuador. And we thank them for their collaboration and with us. So the work in my lab is rooted in biodiversity and an appreciation for conserved and novel physiological and behavioral traits. So many behavioral and physiological traits are shared across animals. So for example, all vertebrate animals have to compete for resources with aggression or participate in reproductive behaviors. And similarly, physiological processes are also highly conserved, like ovulation or spermatogenesis to support these reproductive behaviors. But some traits, though, are evolutionary innovations, where animals come up with these new physiological processes or behavioral patterns to overcome challenges or take advantage of opportunities in their environment. And an example that I'm showing you here of a physiological innovation is chemical defenses. So all these animals have come up with ways to avoid predation and protect themselves, whether it's ants that carry alkaloids for defense, or this bombardier beetle who's shooting boiling acid from his rear end. There can also be many behavioral innovations seen across animals, such as parental care, which has evolved independently across many taxa. So it's not an ancestral trait, it's a derived trait that has evolved many, many times. And so the question that unites my lab is we want to understand how variation in ecology drives innovation in physiology and behavior. So how do animals come up with new ways to interact with their environment, and what are the molecular underpinnings of these changes? A goal for our lab is understanding how ecology drives these changes to influence these processes. And so most of my lab, not all, uses poison frogs as a model system to address these questions. So they have a number of physiological and behavioral innovations that various people in our lab study. So first, they're chemically defended and carry these small molecule alkaloid neurotoxins on their skin to protect themselves from predators. And they don't make these toxins themselves, but they sequester them from their diet of ants and mites. And so they sequester some of these compounds unchanged, but the frogs do have the capacity to metabolize these compounds into from precursors into more potent toxins, which is a process that we'll talk a little bit about. But there are also closely related non-toxic frog species, and this allows us to compare the physiology of toxic and non-toxic species to give us hints at how this trait has evolved. But all species in the poison frog clade also show parental behavior, where they move their hatched tadpoles to pools of water, piggyback style. So I've drawn little tadpoles on the back here. So they give all their offspring piggyback rides to water. And these, because of this transportation behavior, water resources are incredibly important to their fitness. And so several people in our lab study spatial behavior by placing tracking tags on the animals and following them around the forest. And so the choice of water pool, and not only where it is, but the size of the water pool is incredibly important. And some species will place tadpoles in very small resource poor pools. And these species have evolved special modes of parent-offspring communication. And so these are the three stories that I'll be telling you about today, recognizing that all of these traits are tightly linked to the environment. And so we're going to be talking about, the bigger picture of ecology and evolution, but in each story we'll be going down, through behavior and physiology, through neurons and epithelial cells, all the way to proteins and small molecules. All right. So the first story I want to tell you about is a chemical ecology story, where we're linking ecology to diet and neurotoxins. And so this chemical defense trait is influenced by both frog genetics and the environment, because the frogs acquire these neurotoxins from their diet of ants and mites. And these arthropods are very sensitive to habitat quality. And in fact, many studies in ecology use ants as an ecological indicator for forest health. And so much of our field work takes place in these secondary forests. And so secondary forests, meaning that the large trees have already been cut down, which is what most forest left on the planet is. But it's still a pretty high quality forest. There's lots of vegetation, so on. But sometimes we return to our field sites and they look like this. And what has happened here is that the trees have been cut down for lumber and banana trees have been planted. I'm not sure if you can see my cursor, but these plants right here are banana plants. So a lot of farmers will cut down the forest to grow bananas for export to the United States and Europe and globally. And so you'll also notice here that the habitat is brighter and warmer. And so the environment is very different for all the animals that live there now. And so because chemical defenses rely on molecules moving across these trophic levels, we wanted to ask how human land use might be impacting arthropod communities and frog chemical defenses. And so how we did this is we selected a forest edge that had a secondary forest next to a reclaimed cattle pasture. And we quantified arthropods in the leaf litter and frog diets, as well as alkaloids in the ants and the frogs. And so this was work done in Ecuador in collaboration with Centro Ambato, which is a amphibian conservation research center led by Dr. Luis Coloma, a herpetologist. And we also worked with Dr. David Donoso, an entomologist. So all of our field work, international field work, includes local scientists. This work was done by Nora Moskowitz, a graduate student in my lab on the Stanford side. And this work was also done by 2 high school teachers and their 65 high school students. So a cornerstone value in our lab community is community engagement, which is required of all of our lab members. And this study is a good example of our core value because these 2 teachers and their high school students were co-authors on this manuscript, and the teachers came with us to Ecuador to conduct field studies. They collected samples and they brought those back to their classrooms where the students participated in original science. Okay, and so I'm going to describe a little bit more to you how we collected all these samples. So we collected a number of samples in the forest and pasture habitats. This included leaf litter arthropods, which were collected in Winkler traps. And the Winkler traps are an established method in ecology to collect arthropods from leaf litter samples. So you can see the 2 high school teachers hanging their Winkler traps here while we were in the field. And then we also collected poison frogs in both habitats and examined their chemical diversity, as well as the ants. Okay, so we first examined poison frog diet and separated the specimens from the stomach contents into ants, larvae, mites, and then grouped all of others. And we found that overall, diet differs across habitats. So this is an NMDS plot, which you can think of kind of like a principal component analysis. Well, a PCA is actually a special kind of NMDS plot, but the dots that are closer together are more similar to each other compared to the dots that are further away. And so what this plot tells us is that there is significant differences in their diet and their diet composition. And if we look at different prey categories, we can see that the frogs in the forest are eating more ants than the frogs in the pasture. The reason this is important is because many poison frog alkaloids are ant-derived. And so remember, they're getting their chemical defenses from what they're eating. And so what we did next is we compared the species diversity of ants in the frog diets to the ants in the leaf litter. And so in this NMDS plot, I'm showing you these circles, which each circle is a frog. And the data that went into this plot is what the different species of ants that they were eating. And this is compared to these squares, which are leaf litter samples. So we're basically asking, what are the frogs eating? You know, you have this menu of different things to eat in your environment, and what are they actually eating off the menu? And so what this plot tells us is that frog diet is only a subset of the leaf litter, which means that they're only eating a subset of species from their environment. And when we separate out this by habitat, we can see that the species diversity of ants is very different. And so since many poison frog alkaloids are ant-derived, and there was large differences in ant species diversity across habitats, we wanted to pinpoint which alkaloids were coming from which ant species. And so to do this, we use gas chromatography mass spectrometry to ID alkaloids across ant species and colonies. And we found several interesting things, which I'm going to show here in a heat map, where darker red is higher abundance and white represents undetectable. So we weren't able to detect that compound on the machine for that particular sample. So some alkaloids are present in all the ants that we collected. And then there are other compounds like this 223H and 225C that were only found in a few species. So for example, these two species here, Ectotema and Linapithema, had high levels of this alkaloid 223H. And so what we found interesting about this is that these ants are associated with open spaces. So Linapithema is the genus of the invasive Argentinian ant, for example. And so they like open hot spaces. And we found more of them in pasture samples. And so what we wanted to know is if 223H is more prevalent in pasture frogs. And so another way of asking this question is, can we predict poison frog chemical defenses based on the local ant species diversity? And so when we quantified the alkaloids in poison frogs from both the forest and pasture habitats using GCMS, we found that overall chemical defenses profiles differ in the frogs from different habitats. And then second, we found that overall forest frogs have more alkaloids than pasture frogs. And so this is true generally, but I'm showing you here 4 alkaloids as an example. So 3 are more abundant in forest frogs, but I also want to make the case that this isn't universally true. Some alkaloids are more prevalent in pasture frogs. So for example, this compound right here. And these differences don't seem to be linked to chemical structure because these 2 alkaloids at the top are structurally very similar to one another. And so back to our original question, can we predict poison frog alkaloid profiles from ant species diversity? And what we found was that yes, as when we look at 223H, which is more prevalent in pasture-associated ants, this is also more prevalent in pasture-associated frogs. And so it looks like we can do some predictions based on the environment and for frog chemical defenses. And so to summarize what I've shown you so far, I've shown you that habitat quality influences ant species diversity and that frogs in different habitats are eating off different menus. And then what I've also shown you is frogs in disturbed habitats have fewer chemical defenses than so the ones in the pasture had less chemical defenses than the ones in the forest. And the reason why this is important is because it informs our conservation strategies with regards to these species, because when frogs lose their access to their chemical defenses as human land use is increasing and destroys the forest, we need to make we need to prioritize poison frogs that are in locations where they might not have access to these prey items. And so I think this ecological work is important, but the reason we began working on poison frog chemical defenses in the first place was to understand how new physiological processes evolve. And so I'm going to take you on a brief summary of our work investigating chemical defenses from a physiological perspective and asking the question of how do frogs sequester neurotoxins. And so this image shows a model for our alkaloid bioaccumulation. And so as toxins are acquired in the diet, an uptake of alkaloids likely takes place in the intestines. And the final destination of these toxins is in the skin, where they're stored in these skin granular glands and are released in a stress response that's triggered by predation. And so it's likely that these toxins arrive from the intestines to the skin via blood transport, which is routed through the liver. And so the liver is the site of drug metabolism in mammals and many other animals. And so our hypothesis is that the liver is also the location of toxin metabolism as well. So for example, I mentioned to you that some species of poison frogs can metabolize this dietary precursor, 251D, into this allopamiliotoxin, 267A. And this involves these 2 compounds look really similar. And so I'm circling here what the added to the 7 carbon. And this change might seem really small, but it actually increases the potency of this neurotoxin about 5-fold. And so much of our physiological work in this topic focuses on how alkaloid sequestration changes cellular dynamics within a bunch of these tissues. And so most of this work involves feeding experiments where we feed non-toxic poison frogs either control or alkaloid containing prey items. And so in the experiment I'll describe here, frogs were fed Drosophila dusted with a vitamin mix only or a vitamin mix supplemented with 2 alkaloids, decahydroquinoline and familiotoxin 251D. And then 4 weeks later, samples were collected. And so this experiment was actually done by our Ecuadorian collaborators in the Conservation Center, Centro Hambatu, run by Dr. Luis Quiloma. And so 4 weeks later, we collected skin samples from the animals and used GCMS to quantify alkaloids. And what we found is that while the control animals lacked alkaloids, because we didn't feed them any, the alkaloid-fed animals had both familiotoxin 251D, so this is the compound that we actually fed them, and they had the metabolite allopamiliotoxin 267A. And so this demonstrates, we didn't feed them this compound. The frogs metabolized this compound into this more potent metabolite. And so this showed us that these frogs can also do this in captivity. And so then what we did is we used proteomics across different tissue types to quantify how protein abundance changes when alkaloids are present. And so this work was carried out by Stephanie Keaty, a graduate student in the lab. And so across the skin, liver, and intestines, we found many protein classes that differed in abundance between control-fed, which is here in the light green, and alkaloid-fed, which is here in the dark green. And I'm showing you 2 examples from each tissue type here, although the number of proteins that were significantly different in abundance are here in this Venn diagram at the top. And so, for example, complement C3, a protein important in the immune system function, is increased with alkaloid exposure in the skin here and the intestines over here. Solute carrier proteins also change in abundance and might be linked to toxin transport across cells. And finally, there are several cytochrome P450s that also increase in abundance and may be linked to toxin metabolism. So overall, we see a strong immune system response, which Steph is now studying for her dissertation work, and she's asking how alkaloids impact the frog immune system and the microbiome. And she's doing this lovely work in collaboration with a Stanford undergraduate and a community college, a local community college student. Because as Lily said, I run this community college program here at Stanford. I'm happy to talk about it if anybody's interested. Okay, and so these studies are largely, though, correlational, and so we don't yet know which of these proteins actually bind the alkaloids. And so this work, these functional studies into toxin protein binding, is being carried out by Aurora Alvarez-Buya, a graduate student in the lab, along with a Stanford undergraduate, Mary, and in collaboration with Professor John Long. And so poison frogs can carry 10 to hundreds of different alkaloids on their skin. So one individual frog can have hundreds of different alkaloids. And in fact, there's been over 800 different alkaloids described from poison frogs that comprise over 30 different structural classes. And so here I'm just showing you 4 as an example to give you a sense of like structural diversity. And so each class of toxins targets a different ion channel or G protein coupled receptor in the nervous system. And so these are all neurotoxins. And it's unlikely that poison frogs have mutated all the proteins that confer them this resistance. And so instead, we hypothesize that poison frogs have plasma proteins that bind alkaloids for sequestration to the skin and at the same time protects their nervous system from the neurotoxins they're taking up from the environment. And so what Aurora has done is she has found some photo-crosslinking probes that resemble our poison frog alkaloids to functionally identify which proteins might bind our alkaloids. And so these photo-crosslinking probes have also a terminal alkyne for click chemistry and a cross-linking group. And so she's found also several other probes that are commercially available. And so these probes in the next slide will be represented by this cartoon. And so in a series of experiments, what Aurora did is she collected plasma from frogs in our lab colony. And then she adds the probes, which bind to some plasma proteins, but not all. And then she uses UV treatment to cross-link the probe to the protein. And then she adds a dye to be able to visualize the proteins on a gel. And so in this experiment, Aurora tried several different photoprobes that resembled alkaloids. And so the different bands here, or the different lanes, these are different photoprobes, PB1, 2, etc. And then whether or not she had the UV treatment or not. And so this gel not only demonstrates that there are several proteins that bind these photoprobes, but also when we look at this loading gel, we see that, well, not only did she evenly load the lanes, but she also the proteins that are binding our photoprobes are not the most abundant protein in the sample. And then, so once she had this technique worked out, she next did, I think, a really clever experiment that involved competition between a photo-crosslinking probe and a poison frog alkaloid. So it starts the same as before, where alkaloid-like photo-crosslinking probes are added to the poison frog plasma. And then in this experiment, though, she then added a poison frog alkaloid to compete for binding. And so then she UV treated to cross-link the probes. The dye is added to visualize where which probes or which proteins have probes attached. But the important thing here is that some proteins will be tagged when the toxin is absent, but when the toxin is present, it will be absent in the gel because it's competing for binding. And so Aurora found this pattern in some proteins where the protein is not tagged when the poison frog alkaloid is present, which suggests that she's found some poison frog plasma proteins that bind these toxins in vitro. And she's included some other poison frog alkaloids to look at the generalizability of toxin binding versus specificity. And so she's found that this protein likely binds other alkaloids as well. So these are other poison frog alkaloids here, as well as a nicotine control. And so it binds some, but not all of them that are tested. And we're still waiting on the proteomics core here at Stanford to get us the ID on this protein. So unfortunately, I don't have that answer for you today as to what it is. But we're excited to, hopefully we'll find out very soon. So in summary, what I've told you is that we've found that the immune system and small molecule binding proteins are involved in chemical defense sequestration, and our current and future experiments are focused on functionally identifying which proteins bind to the alkaloids, and then knocking them out in poison frogs or knocking them in other frogs to see if we can tune, knock out, or enable chemical defense sequestration in other species. Okay, and so so we've used a mixture of approaches, including ecology and proteomics work to show how poison frogs acquire their chemical defenses. But as I mentioned, there are closely related species that are unable to acquire these alkaloids even when they eat them. And so, in fact, toxicity has evolved many times across the clade of dendrobated poison frogs. And so we're now using comparative approaches across these species to study how this trait has evolved. So this is kind of where we're going in the next couple of years. So in addition to the environment shaping physiology, their poison frog behavior also co-varies with ecological resources. And so, for example, all dendrobated poison frogs show parental care, which consists of transporting tadpoles piggyback style to these pools of water where they can grow up until metamorphosis and they leave the water as little froglets. And so these water resources are incredibly important for frog fitness, as frogs can choose to place tadpoles in very large water pools with lots of resources but higher predation, or in super tiny pools, which are somewhat safer, but they're also resource poor, meaning that there's not a lot for the tadpoles to eat. And so this choice of pool size has led to species differences in who cares for offspring. So in species that utilize small pools, mothers care for the offspring and deposit eggs to supplement nutrition. However, in species where males care for offspring, they utilize these very large pools. And so the neural basis of sex and species differences in parental care is already published by a former postdoc in the lab, Ava Fisher, who's now an assistant professor at the University of Illinois. And so since all of her work is published, I instead want to show published work. And so I'm going to tell you I'm going to tell you this one story about spatial cognition. And so spatial cognition has been studied for well over a century. And so in mammals, males are usually better at spatial tasks than females, although it does depend on the task. And this is reflected by the size and function of the hippocampus. So this is a brain region that's important for spatial memory and behavior. But comparative work in mammals has shown that sex differences are lacking in monogamous rodents. And so this led to a theory that male superiority in spatial performance is an adaptive trait linked to reproductive fitness. But this adaptationist explanation can be countered with the idea that of testosterone spillover, where just having testosterone around is enough to increase spatial performance. So for example, you see trans individuals who begin to take testosterone therapy also increase their performance on spatial tasks as well. So this also happens in humans. And so it's been difficult to test between these two hypotheses, these adaptationist or life history trait hypothesis, and then this testosterone spillover hypothesis, because there are no mammalian species where females have larger home range sizes linked to reproductive fitness. And so this is where our poison frogs become really useful, because we have these species differences in where the sex that cares for offspring, so either mom or dad, and their care of offspring requires spatial tasks of finding and remembering where these water pools are located. And so our expectation here is that the sex that cares for offspring will have larger home ranges. And so what we do is we do all of this work in the field, and we attach tracker belts to these frogs and follow them around the forest as they live out their lives. And so this work was pioneered in the lab by Dr. Andreas Pasukonis, and he has been joined by Daniel Shegovich, a grad student in my lab, and Jenny Serrano Rojas, who's an incoming graduate student. And so these frogs are too small to carry around a bunch of electronics, and so they have to be located with receivers every few hours by the frog tracking team. And so in this set of experiments I'll be showing you, the team established home range size for 78 frogs by measuring the movement of each frog for roughly 2 weeks. And what our question is like, what is the size of their home range, or where they like to hang out? And this work was conducted in Ecuador and French Guiana. And so here are our results for home range size. And I've highlighted here the sex that cares for offspring. And in 2 of the species, home range size is larger in the parental sex, which partially supports this life history hypothesis. And so, but now that we've established that these sex differences in home range size, we next gave the frogs a spatial task to ask if there are sex differences in navigational ability. And so for the next set of experiments, once home range location was established, the frogs were moved either 50 or 200 meters away, which doesn't sound like a lot, but it is a lot for a frog the size of your thumb. And we measured their ability to return home and their speed and their path taken. And so here I'm showing you the results for just one species moved 50 meters to be able to orient you to this kind of data. So home is the gray circle, and the paths taken by males are in blue and the females are in red. And in this data set, males were better able to make it back home compared to females. So females, only half of females made it back home, and most of the males made it back home. And I just want to say that if they don't make it home, we move them back home. So don't worry, we're not like making a bunch of lost frogs in the forest. Okay, so when we look at other species, the sexes were more similar in their performance, but their path taken was very different. So Femeralis over here had a very straight path home, and these frogs are kind of, they have a different search strategy. So they have wider movements, for example. And so when we look at the data at 200 meters, now we see some more pronounced sex and species differences. So in Femeralis, for example, females don't return home at all. And then over here with the Sylvatica, we see both male and females are unable to return. And so the one overall pattern that we can take away from a lot of this field work is that males tend to be more mobile when translocated, and in some species they're better at making home. And so to summarize our work up to this point, we had made predictions about space use and navigation based on the life history hypothesis of spatial cognition, where sex differences in space use and tasks would reflect reproductive strategy. But there were many differences in what we observed, and I've highlighted some of them here in yellow, where there's differences between our a priori expectations and our results. And so it could be that hormones influence navigational ability, and we did collect hormone samples in the field, and Andreas is still working on running a bunch of hormone plates, so these results are still pending. But we're also seeing that we may need some more fine-grained, might need a more fine-grained approach. Because, for example, when you look at, when you break out behavior by what the animal is actually doing, so for example, in Tinctoris, the species here, they didn't have any sex differences in home range size or navigation. But if we separate out what the animals were doing, either visiting a pool or meeting or doing something else, then you can see that this is where these sex differences start to begin. Whereas, for example, males have a lot more movement when they're doing parental care. And so being able to also measure what they're doing as we're tracking them is becoming more important. Okay, so to summarize so far, we used species differences in reproductive strategies to study spatial behavior. And these strategies are linked to water resources in their environment. And interacting with these water pools leads to sex differences in home range size. And this behavior might be modulated by hormones as we see in mammals, but further work is still needed to understand that relationship. So in parallel, we wanted to understand the neural basis of spatial behavior in amphibians. And the mammalian hippocampus, which is marked here in blue, is one of the most well-studied brain regions in mammals. And it's part of the pallium, which is shown there in green. And the hippocampus is the location of place cells and cells that encode velocity and other parameters important for moving through your environment. But mammals didn't invent the hippocampus, just like they did not invent the pallium or the cortex from nothing. And mammalian brain regions and neurons have evolutionary antecedents in other animals where the pallium and hippocampus-like brain regions exist, although their function is not well described. And so, for example, up until very recently, place cells were thought to be unique to mammals until someone bothered to look for them in birds and found that food caching birds do have place cells in their hippocampus. And so what we wanted to see is that what the neural basis of spatial behavior in these amphibians, what were the patterns that we were seeing, and could we correlate them to navigation? And so to get a sense of how pallial brain regions in the amphibian brain might be associated with spatial behavior, Andreas collected a similar set of navigation experiments, and he did a similar set of navigation experiments in the field, where frog home range size was established, and then animals were either captured and released back home as a kind of a sham control, or two groups were translocated 200 meters. One group navigated back towards home but was captured before they arrived, and the other group includes lost animals, so animals that were moving in the wrong direction and they were clearly didn't know where they were going. And so we collected brains for each of these frogs in the field and asked if the medial pallium, which is the proposed homologue of the mammalian hippocampus, had increased neural activity during navigation. And so we also quantified neural activity in the dorsal and lateral pallium as well. And so as a marker of neural activation, we use the immunohistochemical detection of PS6, which is a marker of ribosomal phosphorylation. and neural activity. And the idea here is that when neurons become more active, they need to make more protein, and so they phosphorylate their ribosomes. And so this can be thought of as analogous as to like CFOS, or like a proxy for neural activation. And so what this means in practice is that we're like counting the number of brown cells in different brain regions. And we use this method because it basically enables us to do field neuroscience, as our animals are collected in the field and their brains are processed much later here in the lab. And so I'm showing you the results from this experiment with groups on the x-axis and neural activity on the y-axis, and the different brain regions as box plots. And so we were really surprised when the medial pallium did not differ in the number of PS6 positive cells, or the number of cells that were active when the frogs were doing these navigation tasks. However, the number of active neurons in the dorsal and lateral pallium, so these blue and orange box plots, did increase navigation, just kind of a new role for these other brain regions in spatial behavior. All right, so to summarize these brain studies, we found that the dorsal and lateral pallium, rather than the medial pallium, was associated with spatial navigation. And of course, immunohistochemistry as a proxy for neural activity does not establish or negate these brain regions as being important. And so what we're currently doing is establishing neural recording methods in cane toads. So poison frogs are too small to carry neural recording equipment on their heads, but cane toads are enormous, like enormous. And so Daniel Shakovich in my lab has dedicated his dissertation work to working with these toads, and he is working with Professor Gusung Hong in the Department of Engineering here at Stanford, who developed mesh electronics, and they've implanted them into frog brains. And so I'm happy to report that Daniel has now made like the first ever neural recording from a freely moving amphibian. And so I'm really excited to see where he takes that work over the next couple years. So for the last bit, in my last couple minutes here, I want to take a deeper dive into these neural mechanisms of behavior, and talk about one of my favorite projects in the lab. And so we've started doing these in-depth studies on how brains govern social behavior in these frogs here, which are mimetic poison frogs. And so we have a number of teams asking different questions that I'll mention here, and then I'll just go into one specific example. So poison frogs are monogamous and biparental, and so a couple of individuals in the lab, led by Jess Nowicki, are investigating the neural basis of monogamy. And as I said, they're biparental and share parenting duties, and so a graduate student in the lab, Billy, is studying how parents interpret the needs of their infants. There are many aspects of the environment that influence tadpole behavior, which is being investigated by Lacey Fisher. But the story I'll tell you about today is our efforts to understand the neural basis of communication in tadpoles, which is what, and so I'll tell you about the work of Dr. Julie Butler. And so, but before I get to brains, I want to first explain to you the behavior, because the behavior is a little unusual, well, as in people don't know it exists usually. And it's a really fascinating behavior. And so it's this egg feeding behavior in poison frogs. And so here, moms provide unfertilized or trophic eggs to tadpoles. This extended provisioning benefits offspring, so meaning that they're more likely to make it past metamorphosis into froglethood. And the tadpoles also beg their mothers for meals. So I'm going to show you I hope you can see this video here without it being too patchy. But what's happening is that this tadpole is nipping and vibrating against its mother. This motor display is required to get food from mom. Mom will not feed the tadpole unless it does this dance. And it's more likely to beg for food when it is hungry. And so people have hypothesized that it's an honest signal of a nutritional need. And so some work has shown that these tadpoles fed by moms also carry toxins. But more broadly, it allows for the exploitation of novel habitats. And so this idea was first put forth by Jason Brown and Kyle Summers at East Carolina University. And the idea here is that you can either place your tadpoles in really large pools of water with lots of resources and predation, or in very small pools of water where there's no predators, but there's also nothing to eat. And so to be able to utilize these safe pools, parents had to evolve this behavior to come back and feed their tadpoles with these eggs. So for a nutritional supplement. So a lot of people in the lab are now focusing on begging behavior in these tadpoles. And so this is not unique to tadpoles though. So lots of infant animals beg for food. So, babies beg for food, chick, like you can think of like chicks begging in the nest for food as well. And so here's another video of that happening just to show you what our assays are kind of like in the laboratory, what this begging behavior looks like. And so our question here is, how do new modes of communication evolve? And so the first step is understanding what the neural basis of this begging behavior is. And so the experiments that I'm going to show you were done by Julie Butler. And this was in collaboration with Professor Scott Edwards and a postdoc in his lab, Devraj Singh. They're ornithologists and study begging behavior. And so it was a really nice collaboration for us to get into this work. And so when we first started doing these assays, when we presented a female frog to a tadpole, we noticed that sometimes that they didn't beg and sometimes that they did do this begging behavior. And so we decided to utilize this individual variation in response, where about only 70% of them were begging, to try to take a step towards understanding the neural basis of this behavior. And so here's we actually quantified the behavior on a graph here on the left. And so we use the same approach, so this immunohistochemistry for phosphorylated ribosomes, and we quantified across the number of activated neurons across these different brain regions important for social behavior. And so there's not a lot known about the tadpole brain, but that doesn't mean we were starting from scratch. And so the brain regions that regulate social behavior in mammals are really well worked out. So this includes the dopaminergic reward system and social behavior network. And so they have very typical gene expression patterns that include genes from the dopamine system, sex steroid hormone receptors, and neuropeptide systems like vasopressin and oxytocin. And so one of the things I did as a graduate student that Lily mentioned in the intro is that I looked at the neurochemical patterns and developmental trajectories of all these brain regions in mammals and made kind of like a Rosetta Stone almost of like a map of where all these brain regions were and all other vertebrate taxa to allow comparative studies across the same brain regions. And so this has been also really useful in our work in frogs now. So the brain regions that we're studying are similar in gene expression and function to the ones in mammals. And so I'm going to show you the data on this like spider web plot. And what I want to show you is that overall brain activity, so the further away from the center you are, the more active neural activation we have here. So overall brain activity is higher in begging tadpoles, which is not surprising because they're like doing a lot of motor movement. But still, after statistically accounting for this overall higher brain region activity, we still see that there are a couple brain regions that are more active in begging compared to non-begging animals. And so 3 of these brain regions are associated with dopamine. And so one of these, for example, is the lateral septum. So I'm showing you here the raw data in box plots. So this is the number of PS6 positive cells in the different groups, begging and non-begging. So, and the number of active neurons here actually correlates with the time that animals spent begging. We also see something very similar in this brain region called the posterior tuberculum. So for those of you who do neuroscience,
lottery system.
For those of you who do neuroscience,
Address or mobile number for few.
I think that's me.
Can everyone get themselves muted? Yeah.
Okay. I'll just keep going. All right. Where was I? Oh, animal. Yeah. Okay. For those of you who do neuroscience, the ventral tegmental area and the substantia nigra, which are big dopamine brain regions in mammals, those are like very separate, but in anamniotes, so fish and frogs, that's still just one brain region. They haven't separated out yet. So this is the main source of dopamine in the frog brain, which is also correlated with time spent begging. And then we also see this overall pattern in other brain regions that are associated, that receive a lot of dopaminergic input from the posterior tuberculum. So it's no surprise, for example, that these show higher activation because these brain regions are innervated by neurons from this brain region over here. So then once we identified where begging behavior might be happening in the brain, we then wanted to know what kind of cell types are regulating begging behavior. And so we again use the same PS6 antibody, but instead of doing immunohistochemistry here, we're going to do an immunoprecipitation. And so here we do a whole brain homogenization and take an aliquot of total RNA. And then we also do an immunoprecipitation to isolate the RNA from ribosomes that are from these phosphorylated ribosomes. And so these are more likely to come from these active neurons that are actively transcribing genes, more so in the behavior that we're looking at. So it's a way to get at behaviorally relevant gene expression rather than sequencing an entire brain region. And so I'm going to show you the data here in a box plot, but I want to show you how to interpret this data first. So the dots on the right side are going to be genes that are being expressed in active neurons. So they're enriched transcripts, whereas genes on the left side, you can interpret, so they're depleted in the data set in the immunoprecipitation, and you can interpret these as like silenced neurons or neurons that are less active. And so when we do this, we find a couple things that are differentially expressed. And there are a lot of dots, right, because this is like all the transcripts being expressed at once. But what we were specifically looking for are genes that are neuronal cell type markers. And so some of which I've highlighted here. And so because dopamine was such a prominent, dopaminergic brain regions was such a prominent figure in our brain region data, I'm showing you the genes associated with dopamine signaling here. So these are dopaminergic receptors that either increase or decrease intracellular signaling. This is tyrosine hydroxylase, which makes dopamine. And then this enzyme here converts dopamine to norepinephrine. And so then what Julie did is she wanted to see how dopamine regulates begging behavior. And so she did this co-stain. So she co-stained with tyrosine hydroxylase, which is the rate limiting step in dopamine synthesis, and then co-localized them with our marker of neural activation here, and found that in some brain regions, like the posterior tuberculum, that these tyrosine hydroxylase neurons are more active when the animals are begging. To functionally test the role of dopamine, though, she had to do some pharmacology. And so she injected tadpoles with either vehicle or a dopamine 1 receptor antagonist or a dopamine 2 receptor antagonist. And so what she found is that when you treat the animals with D1 receptor agonists, they're much more likely the latency to beg is much lower. They beg more frequently. And then also because dopamine is an important regulator of motor function in general, she also quantified swimming time, which was not different across groups. So to summarize what I've shown you so far, I've shown you that dopamine regulates begging behavior through the D1 receptor, and we're continuing to investigate some other markers that came up during our untargeted screen of active neurons, our molecular profiling experiments. And so we're now poised to dissect this neural circuit underlying begging behavior. And so I didn't have time to share this with you today, but we've also established that begging behavior is based on olfactory cues, and we've also identified the motor neurons in the spinal cord that are more active in begging poles. And so once we kind of figure out this circuit, we're interested in looking at how circuits evolve and how they're modulated between different species. And so we have two very closely related sister species in the lab, one of which whose tadpoles beg and the other one doesn't. And so we're interested in looking at how these neural circuits underlying communication have evolved. Okay, but more broadly, these parent-offspring interactions reflect these species differences in water pools in the environment. And the location of these water pools then have been linked to species differences in parental care, which we talked about, and are somewhat correlated to movement behavior of the frogs in their natural habitat. And we're also uncovering some surprises on which brain regions are associated with spatial behavior across species, where reproductive strategy does not explain sex differences in spatial cognition, which has long been proposed for mammals. And then finally, the environment is also important for other aspects of physiology like chemical defenses, where species interactions depends on habitat quality. And we're just now beginning to understand how these protein ligand interactions have evolved to allow the evolution of chemical defenses in this clade. And so when I think about, where our work has been and where it's going, I like to think of this quote by Francois Jacob that says that nature is a tinkerer and not an inventor. And we see this in our frogs, and whether it's changing protein small molecule interactions or like shifting configurations of these conserved neural circuits. And this doesn't happen in a tube. It's in response to different ecological challenges like co-opting neurotoxins to defend themselves from predators, or developing strategies to utilize poorly resourced water pools in the field. And so I think our frogs have a lot more to teach us about physiology and behavior, which, after all, are the substrates of natural selection. And so I just want to quickly thank my lab, who is filled with amazing people, our funding. Most importantly, I'd like to thank our South American collaborators. A lot of our work is done in the field. We wouldn't be able to do a lot of the work the way we do. Okay, and I will take any questions that you might have.
Thank you, Lauren. Very impressive. I guess people may raise their hands or type their question in chat. I saw a few hands raised. From Rebecca Starbo. Rebecca, you may ask your question, and I mean for yourself.
Hi, thanks so much for the great talk. I have a question about the first part of your talk, when you're talking about differences in frog toxin composition in the disturbed habitats versus the forests. And I was wondering if you observe any differences in the diversity of predators of the frogs. So like, do you see any differences, for example, in the disturbed habitats where the frogs have less toxin composition? Do you see that there are more predators that would prey on those frogs?
So they definitely have different predators in different like populations and locations. And like different populations also have different color patterns. So like in my frog rainbow here, all these frogs are from they're the same species, but from different locations. And so it's thought that differences in predators in a mixture with mate choice drove these population differences in aposematic coloring or warning coloration that they're toxic. So they genetically bought into being colorful, but so they don't get rid of this, like it doesn't change their color whenever they're non-toxic. And so in disturbed habitats, you are like bright red and not toxic. And so in our particular study, locations were side by side, and so we didn't see any differences in predators. I mean, I think that they have less cover overhead, which means that they would be easier to pick off by birds and things like that. We do know that birds learn to avoid their signal, because once we did a translocation experiment where we brought these frogs from the north down to this like other location, and the birds had never seen that morph before, and a lot of them got picked off before we like realized and were able to like do like some type of like covering. And so their local animals do recognize their signal and avoid them. And so I think the problem is that when they lose their toxins, that like learning process is going to be disturbed, and the animals, my fear is that the animals will not avoid them anymore. Does that make sense?
Yeah, that makes sense. Thanks so much.
Yeah, thanks.
Okay. A second, like kind of similar related to question from Alex.
Hi. So this is a great talk. I love poison frog tropical biology. That was something that I was like really obsessed with as a kid. So my question is kind of getting at like the same topic, but I suppose from sort of the other angle, which is in situations where you have frogs, in secondary forest versus open habitat, and they have this different toxicity composition or just different levels of toxicity. Is there any readout for behavior that's different between those frogs? Are they are they even aware that their composition is different? Do they hide more? Like, are they right? They're these brightly colored frogs that sort of just go where they want and do whatever they feel, but
Yeah, yeah. There's this saying that if you see like a beautiful frog in the forest, that is not an animal that you want to touch. And because usually they're incredibly bold, and they kind of like sit there and display on leaves, like no one's gonna touch me. But I will say, so we don't to get to like more of like the science of your question. I don't think we know really how toxicity changes behavior. So we do know that some of the alkaloids can get into the brain, and that being toxic does change brain gene expression. But how their behavior changes is not quite clear yet. So people in the lab are just now starting to like make toxic and non-toxic frogs and then look for behavioral differences, and specifically in foraging. And so, but I will say that I think probably the biggest differences I see when we're like looking at frogs in disturbed versus forested habitats is just their days are just much shorter. So they're, because it gets, and they don't, the forest is a cool, damp, like darker place. And then the pasture is like hot and it's dry, and so their period of activity is much smaller, so they're out foraging less, and they're also we have seen in our enclosures that they also tend to produce less offspring. They're just out less, and I think that plays into a lot of their changes in behavior, mostly due to temperature. Did that make sense? Did that answer your question?
yes, definitely. Thank you.
Okay, awesome. Thanks.
Okay, do we have more questions from the audience? You may either raise your hand or type the question into the chat box. Okay, thank you, Lauren.
Thank
That
you,
is a very
everybody.
impressive talk.
I just stay here, right?
I don't know if the Zoom link was the same for the round table.
I think so.
Okay.
Oh yes, same room for the remainder of the meetings. The next one's called for 12:45, so everybody has a break.
Yeah, we
and
have.
Lily, I'm going to appoint you host, Lily, before I go, so that you'll be able to control the meeting.
Okay, thank you.
No problem.
Okay. I'll be back in 10 minutes.
Oh, sure.
Okay.
Take a
I'll
break.
be right back.